the Dementia Researcher podcast, talking careers, research, conference highlights, and so much more.
Welcome to the first of our two-part special from ADPD in Vienna. We're going to bring you some of the highlights from the 2025 conference. This is one of the world's leading events for Alzheimer's and Parkinson's disease research, and researchers from all over the world have gathered here today for this week to talk about these diseases and the research that's happening. Hi, I'm Dr Amanda Heslegrave. I'm Principal Research Fellow at University College London, and it's really good to be back to host the show, which I've done previously, and I just need to remember how. So over the next two episodes, we're sharing key takeaways, expert insights, and the latest breakthroughs that have been presented at this year's event in Vienna. So we've got thousands of researchers, clinicians, and industry leaders here coming together to talk about and be a vital hub for collaboration and progress in the neurodegenerative disease research arena. And we're excited today to give you a front row seat. So joining me today, I have Dr Loukia Katsouri from UCL, Dr Imogen Swift from Vespa Bio, but previously from UCL, and Dr Anna Malek from Imperial College London. So hi everybody. Hello. So I'm now going to let you all properly introduce yourself, starting with Loukia. So please tell me a bit more about yourself.
So I'm a senior research fellow at the Sainsbury Wellcome Centre. I work with John O'Keefe, and my main research interests are behavioral characterization of different mouse models of Alzheimer's disease, as well as single-cell electrophysiological recordings in these mice. We have a lot of mice in the lab that we have crossed with tau or APP, and we're also very interested in how tau propagates in these mice, and also how the neuroinflammation is affecting either tau or amyloid.
Mm-hmm. Okay, thank you very much for that. Now, Imogen, tell us a bit more about yourself.
Yes, hello. So, Amanda, you know me very well from 2018 when we worked at UCL, and now I've moved into industry, and I'm at a small Copenhagen-based biotech called Vespa Bio, and I'm a research scientist, so my role is basically to run lots of fun experiments, and it's all related to frontotemporal dementia and Parkinson's disease. So we're a little bit left field for ADPD, but there is some Parkinson's in there too.
Oh no, I think
which
it
is
fits.
fun. Yeah, it fits. Thankfully it fits. It's all dementia related.
Yeah.
But yeah, our focus is basically to target sortilin receptor to treat these conditions, and it's a really exciting time. So it's great to be here though and see lots of familiar faces.
Thank you very much. Now, Anna.
Hi, it's good to be here. I am a research fellow at Imperial, where I recently set up my own group where we're really looking at cell-cell interactions and how they're disrupted in Parkinson's disease and kind of what role that plays in developing dementia downstream. So it's good to be here.
Okay, so that's great. So, right, before we actually start to talk about some highlights, I'd really like to know about what you've actually been doing at the conference. Are you presenting yourselves? And I know you all have been. So first of all, I'll just talk a little bit about what I did yesterday. I was lucky enough to be invited to present at a product theatre for Quanterix. Now, these are a company who produce technology that in our lab we utilise all the time. And so it was really nice to be able to give a kind of 10-year perspective on what we've done with the company and where we started and where we've got to now. So that was great fun. So tell me now about what you've done, Loukia, or doing, or going to do.
So I had an oral on demand, which is on the website and you can watch, and also a poster that was presented yesterday and today. So this is work that started during COVID. So it's a 5-year work. We characterize 2 different APP mouse models at 6, 12, and 22 months of age. We did this longitudinal study with independent cohorts, did some behavioral tasks, about 10 for each one. And then we applied machine learning in these mice to speed up our analysis. Secondly, to remove all of the bias that one experimenter might have against the other. And third, because we want to extract features from this analysis to be able to stratify the animals into each genotype and predict which mice will be more impaired over time or not. So this is quite a big effort from a lot of people. So I'm really happy that it was selected for here.
And one thing is these mice that might perhaps their samples come to us in our lab to do some biomarker
That's exactly
research?
what I'm going to give you. Yeah.
Okay.
As soon as I get the green light, yes,
Perfect.
that's exactly the ones that
Okay,
I'm
well,
going
we'll
to
look
send
forward
you.
to that then.
Yeah.
Okay, so what have you presented or will you be presenting?
Yeah, so we've got kind of two sides of it. There's the preclinical. I've learned lots of new industry words that I try and throw in sometimes. And so there's the preclinical world, and then there's the clinical world. And our boss, Mads Colby, is going to be doing a talk in about an hour's time, actually, on the clinical side, which is very much the world I know less about. But there is biomarkers related to that, so that's exciting. Yeah, exactly. And then the preclinical world is where we've got some cell models and some mouse models, so very relevant discussions there. And we've got a poster for that, which we've been presenting today and yesterday. And so it's been really cool, actually, because it's been work that previous colleagues have been running and now we're continuing with. And it's like lots of very cool assays, which I'm learning more and more about by the minute, because obviously I'm from biomarkers. But we're aiming to increase progranulin levels in these FTD progranulin carriers. And it seems to be very successful so far. And we're in phase 1B, 2A. So it seems exciting. And I've learned what that means now. So that's always good.
Okay.
But yeah, so it's been really cool to be here.
All right, wonderful. So Anna, what are you going to have done?
So I'm going to present my poster. So that's tomorrow and on Saturday. So I'm in the second poster session. And it's kind of a continuation from my postdoc work that I've now developed further at Imperial. So during my postdoc, we generated spatial transcriptomics data, which is really cool data because it allows you to query expression of genes in cells with the spatial component. So you can not just say this is a microglia and this is expression profile, but this is how it changes when it approaches pathology. And so we published this before, but the thing that's been bugging me about generally spatial transcriptomics is that cell segmentation is really difficult. So you throw away a lot of the data you acquire because you don't really know what cell it belongs to. And so what I've been working on since summer and what I'm presenting now is a way to effectively optimize the data that we have and to be able to look at subcellular changes. So pull out the nucleus data that we already have and pseudo-bulk the transcript information. And so we can now start to look at cell states that we know are located to the kind of cell body of a cell, such as specific microglia states are associated with diseases. So this kind of allows us to do some really fun analysis and also look at how cells are polarized close to pathology. So this is kind of a proof of concept for the methods, and we're very excited to make sure it works, it does what it should be doing, which we're slowly confident about, and then ultimately apply downstream to other disease models.
Okay, thanks. That was all good information to have, and I'll come and have a look at your poster later. Okay, so now we're moving on to the highlights so far. I mean, obviously we're only halfway through the conference at the moment, so this is the first half highlights. So I'm going to go round and ask you for some insights and observations so far of what you've really enjoyed. So first we'll start with Loukia.
So the first session that I really liked was one from Rudolf Tanzi. He spoke about translational drug discovery. It was the session Translational Drug Discovery and Experimental Models 1. So he talked a lot about drug repurposing. And the reason for that is because they want to identify targets, like the plenary speaker said, they can't use these repurposed drugs, but they're safe. They could potentially use that to make new drugs, for example. And what he was saying is that they used a lot of transcriptomics and some 3D culture model systems.
Right.
I guess it's from iPSCs, that's what I understood. And he said that especially for phospho-tau hyperphosphorylation, A-beta-42 is very important and is what is driving the tauopathy. So they did some pre-screening. They looked for small molecules to modulate gamma secretase to change the ratio from 42 to 38, which is the protective one, which is really nice. And they found some that changed the ratio and decreased the beta-42 production. They also identified some glioplasticity modulators so that were very interesting. They also used some BV2 microglial cells and some microglial cells from iPSC's neurons. They also made some 3D models. And they did some drug screening with fluticasone and mometasone to see the microglial uptake. And what they also saw, which to me it was very exciting was that Leqembi increases microglial uptake. Yes, which is great. But he said with 50K per year for the drug, it's a bit difficult, but at least based on that, they were able to screen more drugs, and they found about 48 drugs that would do the same to induce microglial clearing, about 52 that changes the microglia to the Roth style and all of this, and 40 that are inhibiting TNF-alpha release, which is also very important. And they found some indications that this is due to microglial senescence, mitochondrial senescence in microglial cells. Okay. So all very exciting. Which,
I'm good. The whole drug repurposing thing at the moment is a huge thing, I think, because there are so many drugs. Actual in-person drugs that we know are safe and are tolerable, but we don't really know all of their effects. For example, the GLP inhibitors.
So huge.
So I think that these kind of talks and this kind of work is really, really exciting. Okay, thank you very much for that.
It's very cool.
Imogen?
Yes, so
first highlight?
Yeah, I really enjoy the poster sessions because I feel like you can really get a lot of information, but also like the key things that you want to ask, whereas I guess with a talk it's slightly different dynamic, and I also am very afraid to go and ask a question at the end of a talk because there's all those people and there's a microphone.
Just try it once.
I've literally never done it. Okay, I need to try it. It's my mission for this week. But one of the posters, it was very relevant to our work especially, but it was Thomas Kukar, who's from Emory University in the US. And he has some really cool work that's actually linking mouse models and the lysosome, and especially granulins on the lysosome. And as my PhD looked a lot into it, I was very biased for my interest in it, but it's actually really, really cool work. And they were basically showing that by adding this granulin 2 and granulin 4, they were able to ameliorate some of the effects of not having progranulin. And so he dived a lot into the lipidomics as well. And they showed that there were decreases in the BMP lipid and the glycosphingosine. Oh no, glycosphingosine was increased. So it was these two kind of lipids that were big. And obviously in the world of biomarkers, I was very interested in that and measuring lipids. And I know that a lot of people are looking at these lipids at the moment and seeing in biofluids and seeing those changes.
So was there any insights on the best way to measure them? Because that's not something that's very easy.
No, so I think they must have done it by mass spec, as in from the poster it indicated. So I know other people do it from mass spec, but I don't think it's easily done.
I don't know of any other way to do it.
No, I don't think there's any
and I also think it's quite specialised in the field of mass spec.
Oh yeah, I think even within that, I think there's 2 people in the world that seem, or 2 groups of people that seem to be able to do it. But it was really interesting to see that the same markers do keep coming up, this BMP. But it was also in the mouse model, and I have seen some kind of controversy around the idea that is actually translatable into humans. So I don't know if that's, because I know when we measure BMPs in the lab and we measured it in urine, we didn't see any difference.
No, I remember you doing that.
Yeah, exactly. So I'm intrigued how that can translate, but also urine maybe isn't the best way to look into it.
But I think about like lipidomics. I mean, there are so many different lipids, and the regulation of them is so
dynamic,
complicated that there must be a reason for this.
Exactly.
And people don't look at it because it's hard. And so we need to do the hard things, I think.
But I think also it's very important to note that the cholesterol in the brain is de novo, so you never get it from the periphery. So
so measuring it in urine is a bit of a waste of time, is what you're saying to us.
maybe it's what is degraded, so it has to come out somehow. But at least the production.
No,
is
that
de
makes
novo.
sense. Sorry, that was flippant.
Just my PhD was on APOE, so I read a lot about cholesterol, and it's so complicated.
I remember I did one of the diagrams to try and include in my thesis, and the diagram got bigger and bigger and bigger, and I was like, I'm going to squeeze this in. But it's very relevant, and like you say, I think now we have dived into a lot of the stuff that is easy to measure or easier to measure, and the sensitive methods are getting more and more sensitive. Maybe it is the time to dive into the harder stuff, but I agree that it's still controversial, and if there's only 2 places in the world that do it, it's still hard.
But exactly.
Cool nonetheless. So,
okay. Well, thanks for that. So, Anna, what have you seen that excites you?
What I found really interesting, I'm switching diseases at the moment from Alzheimer's over to Parkinson's. So I've been, and I think ADPD is like the perfect conference for me at the moment because it's still like the comfort of Alzheimer's. Like every now and again when I feel like a bit like I don't know enough what's going on, I go to an AD session and feel like, hello. But yeah, I've been branching out into the PD session because I really need to know what's going on. So I went to one on PD and Lewy body dementias yesterday about advances in diagnosis, where Vienna from Innsbruck was giving a really interesting talk on biologically defining Parkinson's instead of doing it clinically. So the clinical kind of the way that Parkinson's presents is incredibly heterogeneous, a lot more so than Alzheimer's disease. You have all of these different subtypes. And that makes it really difficult to study it as researchers, but also to run clinical trials. And so there's been a push in the field quite recently to say, actually, we want to define it based on something that we can measure biomarker-wise. So, for example, with the seed amplification assays, by looking at imaging for dopamine loss and combining this with kind of known genetic variants. And that was a really interesting shift to kind of say, this is our biologically defined Parkinson's now. If you have these three markers, a bit to Alzheimer's, what we have already in the AD field, if you have these markers, we can kind of put you into the category of like prodromal Parkinson's or like then actually active Parkinson's disease. And I thought that was really interesting to see concepts that are familiar and that, you know, dealt before an AD being applied now to Parkinson's, with at the end coming to a conclusion that really what the field needs is good PET biomarker for alpha-synuclein. And I was shocked that didn't quite exist yet. Maybe I was very naive, and I was like, this is crazy. There was another talk here at ADP that presented something that could be used as a PET marker for alpha-syn, but that's maybe my next highlight to discuss later.
You know, and I know that alpha-synuclein is a field that there is a desperate need for some good marker that I mean, there are the seeding assays, and that's fine, but it's not enough. There needs to be more. But also, I think that when you talk about the biological definition and the clinical definition, in Alzheimer's at the moment, that's causing quite a lot of controversy, because in the States, they will say that a biomarker positive person, you can diagnose with AD, even if they don't have any clinical symptoms. Yeah. That's kind of... but the European view is that you are now at risk. So, but in Parkinson's, it's going to be even harder because of all the subtypes and all of the kind of, I mean, I don't know enough about Parkinson's about where that, because we know with amyloid, you can have that in your brain and never actually progress. You die before, you know what I mean? You get old and die before. What about Parkinson's? Can that happen with that? Can you have alpha-synuclein aggregates in your dopaminergic neurons and not get it? I don't know.
So I think you can, to some degree, have some alpha-synuclein aggregates, like Lewy bodies.
Yeah.
But I think, right, in Parkinson's, it's this loss of dopaminergic neurons in the substantia nigra, right? And that's, exactly. So that's it. Also, the way the disease presents is quite different. I think the worry you always have with Parkinson's in a way is because pathologically, it's also, there's a lot of heterogeneity. You have a lot of copathology, which I find really interesting, this idea of you have alpha-syn aggregating, but you also have amyloid aggregating, and you know, do we know what's happening there? Like, is the alpha is it amplifying the disease? Is it driving us towards something? All still unknown. Really interesting questions. But I do agree that there's. I think in a way, and this is what we've seen, for example, with Huntington's disease, and what Sarah Tabrizi has been doing, of kind of saying, if you are at risk of developing Huntington's, if we know this, then you can classify you as like stage 1 Huntington's, and that makes then clinical trials a lot easier. So part of me thinks if we can, you know, if you if you re-stratify, might make life easier down the line for
and that actually, that's a really, really good point that was not in my consideration at all. But yeah, good point.
I think it's the benefits of genetics as well, and that, like, the GBA mutation, I imagine it's probably easier to stratify that group for Parkinson's versus the sporadic group.
Yeah.
And
you have
a huge sporadic group. Yeah, exactly.
And I think, it's more info about, what the risk factors are. Okay, Loukia, what's next?
So I'm going to switch gears back to mice.
Okay.
So I watched a talk from Frank Lafeurle that he was presenting some of their Model AD works. So Model AD is a very ambitious NIH-funded consortium of quite a few universities that are trying to humanize a lot of the genes in the mice and then see how they behave. So what they did, they made mice that carried the H2 haplotype of MAPT, which is the protective, I think. And they crossed them with some amyloid mice that were floxed, so you can activate them wherever you want. And also with the human ApoE4, which we know E4 is a risk factor for Alzheimer's disease. And they did some studies in these mice, and they saw that even at young age, at 4 months of age, these mice have plaque formation. They did plaque-associated amyloid and oligomeric diffuse amyloid. And they saw that when you cross them with the E4, the amyloid moves from the parenchyma to the vessels. So you have a lot of CAA in these animals, which resembles a lot of what they see in the humans.
So did they have both the protective and the ApoE?
They had the E3, which is the canonical.
Okay, so it was three different groups, not in the same mouse.
2 groups, E3 versus E4. So to see the normal, so E3 is like human E3, which is it's not the protective. Protective is the E2, and they didn't present any data on the E2 on this.
Rare though, isn't it? Would it be?
It's quite rare. And also you get familial hypercholesterolemia, so with E2,
With
so
E2?
yeah.
Okay, so it's like good, bad.
Exactly. yeah. So exactly. And what they saw is that there was a lot of microglial response to the dense deposits, but not to the diffuse ones. And especially they had a stronger astrocytic response in these E4 mice. And the astrocytes looked weird. They were more kind of like less mature than the normal E3 mice. And they also saw that basically, these mice develop age-related extracellular A-beta deposits and CAA in the appropriate brain regions. Introducing ApoE, it is promoting CAA. Interestingly, they found phospho-tau 217 by staining, accumulating in the dendrites in the CA1 in these mice. And there's a limited evidence of plaque-induced increases in tangle pathology. I guess they're going to look at these mice at an older age. And he also presented some data from old breeders. And he said that they have differences between male and female, as in the female had worse pathology, especially this E4. But what I said to him is what I've noticed in my mice, if you compare breeders with, for example, female mice that never gave birth, the pathology is always worse in the breeders. And he said, we're looking into this, and they're waiting for some naive virgin mice that they haven't bred to compare them. But it's also going to have a lot of repercussions in...
So that's a real kind of hormone-driven. You must.
It seems so that it's bad news for
a good reason not to have children.
Yes, I think it is protective for Alzheimer's if you have no children. But
I wonder in the context of humans if it's shown.
I think it's the hormonal, and I think there was another study I was reading recently that the burden of pregnancy on the body lasts for like years after you gave birth. So I'm not surprised that there's effect on Alzheimer's.
Yeah. Well, I mean, and the whole hormone effect as well. There definitely is something. I mean, they keep doing the studies of the people on replacement therapy.
Yes.
And they keep coming up because it's kind of retrospective and it's not been planned. I don't think that they're coming out with strong enough, concrete enough conclusions from this, because they're looking at a population where they weren't really caring
for
about.
A little
they didn't
bit,
plan to work on it because it's women.
Yeah.
Is that controversial to say?
Yeah, no, it's true. It's unfortunately an under-researched world of women.
Yeah. But I think definitely now that you have more women that by choice don't have children would be a good time to measure this, because
I mean, we know the birth rate across the world is going
declining.
down,
so
Okay. Well, thank you. That was really interesting. Imogen, what do you have?
So I am going back into the biomarker space. I have a safe space, as you know.
I like it there. It's fine. You stay there.
But it was actually interesting. It was a talk yesterday, and It's this group in Rotterdam. I actually used to work with them a bit, so again, slightly biased. But it's the GEMFI cohort, and they've done O-Link on GEMS. So
wow.
I'm always intrigued when people do like O-Link or SomaLogic or these like big mass spec panels, because there's a question of, do you see the usual targets that we know, but also how similar is it? How replicable is it? And it was interesting. They did a lot of, it was actually a lot of data. They also had a discovery cohort and a validation cohort, which I thought was pretty strong. The genetics of the validation cohort was limited by numbers for kind of fair reasons. The genetics for, this was in FTD patients, I should add. So, well, pre-symptomatic and symptomatic, so not just patients. And they had a few markers that flagged up, and neuropentraxins seem to be this. I feel like they're a bit of a theme so far. I know a couple of talks have come up. Yeah, and one of my later highlights also references them. so watch the space. Yeah, a teaser for later. But I think it's going to be really interesting using these panels in the context of studying people and introducing people to trials, but also thinking about diagnostics and things. But I do question the methods a bit if we're getting all different results from the same samples.
I wonder, so what, was it 5,400?
It was 2,900. Okay. It was the one from a few years back, I think. So now it's got even bigger.
Well, it's got even bigger, but I feel like the more studies get done, the more they're finding that the same set of results occur.
Yeah.
So is it necessarily better to go bigger and do thousands and thousands when actually the ones that come out are always going to be not the same, they're not exactly the same, but some of those are in the panel for I'm not saying no reason, but I think that there needs to be some sort of thought about what you're measuring and how useful that is.
It's the risk of the fishing trip, I guess, versus the targeted side. And I think that there's value in both, but I think if we keep repeating these and we keep getting, like you say, similar results, is it just down to abundance of them? And is actually, and what's the meaning behind it? But I think the best thing now is to get more stratified cohorts in all these conditions to really understand what's going up and down, and then we can get immunoassays in, measuring them better. But it was an interesting discussion as well about referring to them as genetic or protein, so basically obviously they're proteins or peptides or however they're measured, but they referred to them in the genetic context with the gene names, and this caused some controversy after, because I suppose you're not really allowed to call them by their gene names because it's not an expression, it's a protein level. So that was also a separate discussion, but I found that interesting. So, but very biased to biomarkers, obviously.
Was that a European cohort or?
Yes, it's this genetic FTD cohort, but it is led by the Erasmus Centre. This Julie Dahab, I think she's led by Harro Seelar, is the leader.
So I'm guessing it's predominantly Northern Europeans.
Yeah, with some Canadian cohorts. It's a bit European with Canada.
Which would be interesting to see if you expand, for example, these biomarkers to Asia or to Africa, how different it's going to be.
Thinking of India, the DRI recently announced that their partnership to make a brain science centre in India where they've got lots of samples. So what they need to do is start to profile these samples and then start proper studies so that because apparently, I learned this morning, 1 in 6 people in the world are in India.
Wow.
Oh, wow. I know.
I know they had a big population, but I didn't realise it.
It was an early morning meeting.
It was very early, but I'm sure that's what I heard. So
it's thinking
about it.
I think that there is such a drive at the moment to look at those
for India and China.
Across the populations. I mean, because... there are, people have shown differences in was it Black Americans and non-white Hispanics? I don't know if that's right, but there are differences in the way that tau accumulates even. I don't know why that
even the GWAS studies have reported very different hits.
In Africans, the ApoE4 genotype is much more common, but accumulation of amyloid is much.
It's not the strongest hit for Alzheimer's disease. Actually.
Exactly.
Fun fact, I learned that dinner last night.
So the things that we learn, that we take for true, are not true.
Not for everyone,
if it's not me.
That is a lot of the issues, I guess, the demographics of many of these cohorts just aren't representative.
And I suppose also then the conferences like this, it's the people who can afford to come. And the people who can't, who have the differences in biology that we need to be looking at are not here. So
Yeah.
now
Progression is needed. I agree.
Yeah, definitely. Okay, Anna, what other things have hit you? Grabbed you?
I mean, it's
Sure
a
no one's
very
hit
intense
you.
conference. It might have hit me as well. So obviously, I'm going to switch back to PD. So I went to a really interesting talk this morning from Amit Kumar from the Karolinska Institute in Sweden, where they've been trying to assess a neuroinflammation, specifically astrogliosis in Parkinson's brains using PET tracers. So they used 2 different PET tracers of very intense names that I was like, it goes over my head. But effectively looked at how kind of astrocytes become reactive in Parkinson's disease, which is really interesting because up until now we haven't. I think there's still an underappreciation maybe in the field for astrogliosis in the field, and found that. Astrocytes or GFAP positive cells on the brain of Parkinson's patients express this other protein ICAM, which is linked to, which at the end we're trying to link to like phagocytosis of dendrites. And so at the end they pulled out this really interesting like GFAP astrocyte microglia crosstalk around dendrites in Parkinson's disease. Where they were starting to hypothesize whether astrocytes reacting might invite microglia to phagocytose synapses, and ultimately ended up with this conclusion of that there were different kind of waves of astrogliosis in Parkinson's disease. So you have this initial wave as the pathology is aggregating, which you can, in post-mortem samples, you kind of have to go quite high into the cortical areas for you to even catch this first wave. And then this subsequent second wave of astrogliosis quite later. So I thought it was quite interesting to see how kind of, I think what I'm interested in is slowly starting to become important to the field as well, this glia crosstalk and how is it regulated, and is it quite similar to what we see in Alzheimer's? Is it ultimately the same cells interacting with each other through known pathways, just in response to slightly different pathologies?
So I think that I'm seeing quite a lot of interest in astrocytes at the moment. That's kind of, it goes in waves with these cells, because I did my PhD on astrocytes and like I was really interested in them, but no one else was. So. But now I see such a lot, but now I see an appreciation of all the cells. I mean, oligodendrocytes are going to come into their own shortly, I believe.
They will. It's like they've been like dancing around the edges. I think the problem is they are really complicated in of itself.
Oh, the harder something is to study, the less it is studied. And we know this by talking about lipids earlier. So, but the more tools we get and the more kind of more people thinking outside the box, and what maybe is important isn't necessarily going to be very easy, but we still need to do it.
Are oligodendrocytes something that can be cultured easily? I feel like is the wrong word, because culturing cells seems impossible either way. But I know microglia and astrocytes are harder. Or is it astrocytes?
Astrocytes are the easiest.
Oh, is it microglia then?
Microglia and neurons are harder.
With microglia is that since they're immune cells, you can't really grow them in stuff like penicillin or streptomycin. So you grow them without any antibiotics.
And just hope for the best.
Yes, very much. That's why I did my PhD.
Just don't breathe on them.
It's like, don't look at them breathing. Looking at them funny. Like, oh no, I've become activated or infected. You're like, I think when it comes to oligodendrocytes, because you have myelinating oligodendrocytes, and then you've got not myelinating oligodendrocytes.
So there's even subtypes.
There's subtypes, and I think I haven't tried it yet, but from what I've like talking to other people, I think the myelinating oligodendrocytes you might just about be able to culture them. But I think the non-myelinating ones are really,
I was reading something about this the other day. And I've got no further insights.
But it's a topic that's relevant to the field.
Yeah.
That's very cool.
It's definitely a topic. So yeah, wow, that's really interesting then. Thank you. Do we go around again?
Do you have any highlights?
Yeah.
That's really not fair. Question. I told you I didn't go to.
I feel like your talk yesterday about saying 10 years with Samara, though, I realised that's been so long. I didn't realise.
10 years since we actually got the machine, and then it took another couple before we started to publish.
Yeah, so the HD1.
So the HD1 was where we started.
I do feel like, I don't know if it's just me, but obviously I have a biomarker hat on, but when you go down to the exhibition hall and a lot of the main like areas are like Quanterix and Alamar, it's really a biomarker. And I feel like, 5 years ago, you would see maybe one person from Quanterix, or like it really wasn't.
They grew a lot.
Absolutely not. And like the interest still, I mean, the buzz around Alamar is quite something at the moment.
Agreed.
And the fact that the CNS disease panel is coming out in mice, actually, surely.
Yeah, well, that's what you need, is what you need across the spectrum to really look into it.
Yeah.
And there will be, I think, more exciting things from there. But at the same time, the Quanterix, the HDX, it's not going anywhere. We're still using it.
Proven to work very well.
On a daily basis.
So do you think that one is better than the other, or do you think there's a lot of fuss about Alamar that...
No, I think that there's different things you can do with them,
obviously.
but you're not going to have one of them that does everything that you need.
Yeah.
I mean, the targeted panels are good, and we need more of them. But I think smaller panels, that's where we're going to end up. I think we're going to end up with smaller panels that will be able to differentiate. We'll get alpha-synuclein sorted. We'll get TDP43 sorted. Yeah. We'll be able to at least exclude some diseases with a panel or include into a further screen. Yeah. And that's got to be better than where we are now. I mean, it's lovely. What was I just going to say? It's lovely if you've got Alzheimer's. I was not going to say that. What I was going to say is that if we could prescribe in the UK and you go to your doctor and he says, oh yeah, you've got amyloid in your brain, but you've also got, like, it's not pure Alzheimer's, so you can't have that drug. I mean, there's very few people who can have the monoclonal antibodies at the moment. I mean, but I mean, I think most people have copathology. And they've got some vascular involvement probably. And so it really, at the moment, we're at a point where a very select few people could have this therapy. Yeah. And those people, I think, are going private right now if they can.
Yeah. But I guess that's also up until now the way we've been studying these diseases has been in such clean, clean inclusion and exclusion criteria. You know, like in Alzheimer's, you model amyloid beta and tau, maybe.
At best.
Together. But like also when you then go into human postmortem brain tissue, you exclude everyone with copathologies or CAAs. And
it's interesting. When I was at Imperial, I used some of their brains to test my base antibody. And as it happens, I saw increased base 1 at the cancer brain. Okay. And I never followed up, but I remember talking with a Steve gentleman. He said, oh, this is really interesting. Nobody has really thought of looking base 1, which is the beta secretase gene, in a cancer perspective. So we don't even know what is happening with these enzymes in comorbidities. So are they going up? Are they going down? What they're doing? So
isn't it? Was it the gamma secretase inhibitors?
The beta. Yes, the gamma.
They did work on, and then discovered that they were affecting so much more in the body.
Yes, because
actually you couldn't target this. Like they were trying
because they're affecting all the notch pathways. So causing cancer.
So So I suppose that the way that you have to start looking at something in isolation. Otherwise, where do you start? Yeah. But then we need, and I think that there will be. So the trials that have happened so far are very selective, very much homogeneous populations. But I know from talks and things that are being planned right now that there's much more of a push to get populations that are just normal populations into all the trials, and so we need to work towards this.
Isn't that one of the NHS initiatives that are following these people over years from a very young age throughout their life? I can't remember.
Is this Our Future Health? No,
is that it? I heard of it when I was in.
It might be.
Is it the UK Biobank?
Maybe that they're selecting samples and they're following them through.
Oh, you mean the 1946 study?
No, that's the old cohort.
Oh gosh.
No, this is one that started more recently.
Maybe it's Our Future Health then. I think it might be that one.
Probably.
But I also think that cohorts like that and UK Biobank are very self-selecting. They're people that decide, I'm going to go and do this. Yeah. Now, I'm going to suggest that might not be representative of the whole population.
Probably.
So, yeah. So I think that we still need new ways.
But there's also,
to
from the
find those
biomarker
people.
perspective, isn't there the big push to really cover as many people as possible for like the work that John Shot is, and obviously with you guys.
The blood biomarker challenges.
Yeah.
So there's the readout study and the adapt study,
Yeah.
and both of
That would
those
be
are
really
really
key.
trying to look at community rather than people
Kind of
coming into
university.
UCLH
Yeah,
or
exactly.
Queen Square or somewhere like that. So there is a real push, but I mean it's never going to be perfect because we're not, as a whole population, all able to do these kind of things. And that's partly why I don't know if you've seen anything. I don't know if I've seen anything presented on finger pricks today.
Oh, right, with the remote testing. So I know that
remote
Quanterix have
testing.
obviously released this new dried
they've released.
blood
They've
spot.
released a method, a protocol for that, which is really exciting.
So is that the key Alzheimer's?
The one that you did at home.
Yeah, this. I mean, potentially this is done at home.
What, Drop AD?
Drop AD is what Michael Scholl is.
Is it
Nick
Scholl?
Ashton and Michael Scholl? Anyway, some guys.
It's really cool, but I think the Drop AD is focused on what the tau and AD. I'm not sure what their focus is.
I mean,
but
I think that what they need or what they want is neurofilament light and phosphorylated tau.
That makes sense.
And if you can get a genotype somehow, I think you can get a genotype actually. They're moved from plasma now. You can at least, I know you can do it on the Alamar platform.
I mean, they do the Down syndrome screening of the baby from the mom's blood. They can detect the DNA of the fetus.
Oh, really, from the blood? So not without the amniocentesis?
Yeah.
Oh,
So
wow.
now
That's good.
It's a few years. Yeah. So I'm sure they can get genotype from your plasma if they can do that.
But I think that, the push towards getting these remote testing things out is going to be, could be transformative as long as we can prove that what we're measuring is what we think we're measuring and that it's
a golden question.
Indicative of brain health. I mean, which is pretty amazing, isn't it? A little finger.
It is. It really is,
finger
actually.
to do that. Yeah.
This actually reminds me of a discussion yesterday because it was Juan Fortier. I'm definitely not pronouncing that correctly.
No, I'm sure
but he does a lot of really cool work and looking at Down syndrome and the links to AD, obviously, because there's a massive link. But he was saying, obviously, with this, all the drugs that are out in AD at the moment and the ApoE4 exclusion element, he was like, do we want to start pushing the idea of screening people, basically everyone?
Yeah.
And is there an extent of how far we should go to get genetics on people so then we can advise, but also then there's the, I don't know, people's information and who, like, you don't want to go too crazy with it. But
if you screened babies for ApoE4 and then you were marked with a...
that doesn't feel ethical.
Genotype
you stop.
Exactly. So it was a really interesting kind of almost ethical debate at that point of saying, how is this going to come into it? But like you say, if we've got such clean trials and such specific cohorts, it's never going to really work.
Or in a way, like we said, we've had these clean cohorts, we've got these clean trials up until now, and let's have this push towards looking at, for example, the Michael J. Fox Foundation is now kind of relaunching a new ASAP call to kind of fund Parkinson's researchers, and one of the themes is on copathologies. And I'm like, this is brilliant. Let's pour some money, let's pour some real money into these things we've been ignoring up until now. I
think that's been a big thing, especially in the research field. I feel like ALS and FTD is an example I had where they weren't fields that overlapped. I mean, they overlap hugely biologically and with the patients and C9 and everything, but the researchers don't overlap at all, really. They don't meet at the same meetings, and now they do more and more and more. And the fact this is ADPD is a really good sign. I feel like more needs to be done for that.
Yeah.
Exciting.
So do we have any more highlights? Having made sure that I didn't have any?
You're leading the highlights.
Amazing, right. But I was going to say, thank you. I was going to say one thing though about the recent trial where they've been using the monoclonal, I think it's donanemab, on pre-manifest
pre-symptomatic
FAD.
Really? Okay, that's interesting.
And I feel like there was a certain amount of success with that slowing of disease onset.
The ideal place to do it, right?
Which gives us real confidence that they can put that into sporadic, a normal population without any, they still need to make the drugs better so that we don't get ARIA and things like that.
Yeah.
But, and maybe by the brain shuttle method of giving the drugs.
I saw this cool talk yesterday. It was these guys that they had like a bimodal antibody that was it was binding to the transferrin receptor, and then it was going into the brain, and then it was targeting, I think it was TREM2, I might be wrong. But anyway, it was really nice because they tried different fragments of that antibody and then a nanobody. And the nanobody wasn't as good as the huge thing, but still it had an effect. So again, I think that would be a nice way to get them in and not having them huge, because I think the nanobody was like 40 kilodaltons, so quite small, whereas the big antibody was 240.
Yeah. Okay.
So
So better methods to get the drugs in, and also drugs that aren't so big, invasive.
Size is obviously a big thing.
Yeah. Sounds good.
I think one other final thing, if we want to do a fact, just because I teased it earlier and I realised that I should probably mention it. I don't know loads on it yet because the talk is technically tomorrow, but it's on this neuropentaraxin 2 as a Samura assay. And to me, I was like, that's super exciting, because I think so far I've only seen it in a mass spec context. I think there was an immunoassay a few years back, but I don't know if it's been replicated.
I've not seen it used in.
We haven't used exactly. It. Well, this is what I was thinking. I was like, it definitely wasn't in our spheres at least. But I think I know the person who's developing it, and it seems really exciting. But also these ratios linking to neuropentraxins.
Yes, I saw.
So there's like tau, and I think that's also being presented on Saturday. So I think it's kind of a, I don't know if it feels a little bit of a bubble of neuropentraxins right now, but I thought that was exciting.
I feel like I might try to get that on demand.
Yes, definitely.
I think I have one last thing from Wendy Noble.
Oh, I listened to that talk.
It was
I was
amazing,
on my
right?
list, and I was like, no, I'm not going to hype about Wendy Noble because I feel like I do that way too much. So now, if you're opening the debate, start for it, and I'm going to chime in.
So basically, the one thing that she said, she got brains, she made iPSCs, did some proteomics, I think, or transcriptomics. So, and she found 2 signatures in these brains. So 4 brains were more like Brack stages. They did long-read sequencing, or they're planning to do. So some of the brains had more seeding of endogenous. To mention that she created astrocytes from these iPSCs, whereas others were more like clearance of tau. So, and she showed like different signatures between these brains, and also very distinct from the control. And she kind of like intrigued me, asking, how is this working? So I think she was spiking the media with some insoluble material. So I'm guessing this is mostly tau.
Yes, I think they did a mass spec, and tau was like the 3rd most abundant protein. There's a lot in the insoluble fractions.
Yes. So I don't know how you feel. I mean, so she also showed that she can get tau when she's injecting mice peripherally. I don't know how exactly that goes into the brain. And she sees a lot of pathology into hippocampus and amygdala. So it intrigued me. It's kind of like the 2 signatures.
Exactly. Also this idea that, if you have AD patients and you get the insoluble fractions, that there are still differences, like there are different phosphorylation sites of the taus, and that they ultimately then lead to different astrocytic responses.
Yeah.
They had the same IPS astrocyte lines, and the same cells effectively got these different fractions, and then either showed some self-seeding tau activity, or not.
Or clearance.
Or clearance. So I think that, again, really makes you question, like, how are we modeling, like,
well, that's the thing.
How?
And Virginia Lee mentioned in her talk yesterday that what they do, for example, when they do their seeding assays in 2 mice, they get 1 brain, they characterize it really well with seeding assays, with the profile in mice, I don't know what, and then they carry the study. And because you could have different responses if you use a different brain, for example, which again is like, what are we even modeling when you do this tau brain extract? So very, very interesting. So so now she's getting more and more into this transcriptomics with long-read sequencing. She said they have an excellent facility at Exeter. So I would be really, really thrilled to see what is coming out of this.
Yeah, they also do some nice, like, organoid co-cultures, and they had a new lecturer who started there a year ago, who kind of has all of the the TREM2 mutations and the TREM2 signaling pathways, and it's like, the Exeter's building, I think, a really nice neuroscience center at the moment.
I went to university in Exeter, so this is very good to hear.
Yeah.
I feel like there was no neuroscience when I was there.
No, but
No, I
they've
think, yeah,
made a
They
big
really,
push. Like
it
they've
used
just-
Yeah.
to be diabetes that was
No, there's
amazing,
also electrophysiology. They recruited Andrew Rundle from Bristol a few years ago.
Oh,
He went there
really?
with a huge group. Yeah.
And I think Katie Lunnan from
Yeah.
the same kind of sphere is
Yeah.
talking tomorrow.
Yes.
I've seen her somewhere. And I was like,
no, I've seen
Yeah.
that.
oh,
Yeah.
no, but
Have I
this
seen
evening after maybe.
Is it?
And I think we've got a
Okay.
transcriptomics session. I was like, I'll be there. Thank
Okay,
you.
Yeah.
I think
amazing.
maybe that's for Katie speaking.
And I grew
Yeah.
up near Exeter. So
Yeah, I feel
Yeah.
like there's
maybe
some links.
maybe I go back. Thanks so much for that. But we don't really have any more time today. If you want to see more from this event, there are lots of posts on social media. Take a look at hashtag ADPD 2025 on X and Blue Sky. You can also visit the event website on keens.com, adpd.keens.com. So thanks to my brilliant guests today. That was a really nice conversation. So thanks Imogen, Loukia and Anna. And of course, the ADPD committee for making all this possible. I'm Amanda Heslegrave, and you've been listening to the Dementia Researcher podcast. So bye-bye.
Bye.
Thank you. Bye-bye.
Bye.
The Dementia Researcher podcast, talking careers, research, conference highlights, and so much more.